Welcome back. In this series of videos, we're going to start looking at some simple Mendelian genetics. This is pretty fundamental stuff, but there are a few points where you can go off the tracks a little bit, so we'll focus on those, and there'll be some additional videos which will give you the opportunity for some practice. So, when we think about simple Mendelian genetics, we're thinking about the inheritance of certain characteristics. We'll come back and define characteristics a little bit later, but we're talking about the things that get passed from parents to their offspring, and then from their offspring to the offspring. So, for example, from parents to their sibling, to their sons and daughters, and then to what will be the grandkids of the parents. So in genetics, there's a specific language we use to talk about the generations, and this language is really only important because it lets us communicate effectively when we're talking about the genetics and the inheritance patterns that we're going to look at. So it's what we'll often be talking about the P, the F1 and the F2 generations. So the P generation are the parents, and so the offspring of the parents would then be called the F1 generation, stands for filial one. And then the offspring of the offspring would be the F2 generation. Now the important thing to bear in mind about these generations is they are all relative. You yourself may have children, and maybe you don't, but if you have children, then you could be the P generation. But your parents relative to you would be the P generation, and you would be the F1, and then your children would be the F2. So bear in mind that these relationships are exactly that. They're relative. So they're relationships, and we have to put them in the context of the other family members that we're talking about. So we'll be talking about the parents, and then the offspring of the parents, and then the offspring of the offspring, the F2. We're also going to be spending a lot of time talking about genes and alleles. And so it becomes important to discriminate between a gene and an allele. So here are two robots. And so the robot on the left is maybe a robot you recognize, it's Wally. And then the robot on the right is his girlfriend, whose name totally escapes me every single time I do this. And so robots are a classification. They contain a number of different variations, and so Wally here is kind of acting as a metaphor for a PC, and then his girlfriend is acting as a metaphor for a Macintosh computer, and you can see the comparison that's being made, and I'll leave you to mock that as you will. So when we think about the classification of things, we're thinking about genes would be robots, and then the variations within the robots would be the alleles. So an allele is simply a variation of a gene. So Wally is a variation of a robot. So here, robot is to gene as Wally is to allele. So we'll talk about genes, and then we will discriminate, and we'll talk about variations of genes called alleles. And it's really the alleles that we're interested in, and it's the alleles which become very, very important when you go on to talk about population genetics. Now, genes and therefore alleles exist at specific locations on chromosomes. So there's a region of the chromosome where you find a sequence of nucleotides, and that sequence of nucleotides produces a functional product, maybe an mRNA that gets converted, the information becomes converted to a protein. Maybe we're talking about a tRNA, or a ribosomal RNA, or a microRNA, all kinds of different functional products encoded by genes which have specific locations called a locus on a chromosome. And you don't have to worry about too much about the details here with all these letters and numbers. All you need to know is that chromosomes exist at specific points on chromosomes, and those are called the locus. So let's go back to thinking about alleles. We're going to talk a lot in the coming videos about Mendel's experiments. And there's quite some controversy about Mendel's experiments and his findings. We'll come to that in due course. One of the things that Mendel helped us understand was that some alleles are dominant to other alleles. And this is one of the places where we often find that students have struggles. Sometimes a dominant allele will be the allele which is expressed relative to the non-dominant allele, which we call recessive. And so sometimes the recessive allele won't be expressed, but that's not always the case. Sometimes both alleles will be expressed. And remember, there'll be two alleles, so I'm going to say both alleles. There'll be two alleles because we're going to be dealing with diploid organisms. And so we'll be looking at pea plants, and then we'll do a little bit of human genetics towards the end of this video series. So you'll always have two alleles. We'll always be thinking about two alleles. And so if you've got two different alleles, say we've got the gene T, Just a name, it's just a name, it's just a noun, it's nothing special or interesting. It's just a gene called T. We could have an allele which we call big T, uppercase T, and an allele we call lowercase t. And the convention is that the uppercase variant, the big T, is said to be dominant to the little t, and therefore the little t is recessive to the big T allele. So what does this actually mean? What does dominance mean? It means that if you have one dominant allele out of the two you could possibly have in a diploid organism, you will see an effect of that allele in the organism's appearance, its behavior, the things it does, its biochemistry, its physiology, all those kinds of things, and we'll talk about those things in a moment. So if you have a dominant allele, even if there's a recessive allele, you're going to see the effect of the dominant allele. Now, if you put two recessive alleles together, that's the only way you can see the effect of the recessive allele. So you can think about the recessive allele being masked by the action of the dominant allele. And you don't need to worry about what these actions are right now, just that if you've got a dominant with a recessive in a diploid organism, you're going to see the dominant allele's behavior. You'll see it manifest in the organism. The only way you can see the recessive allele is if you put two recessive alleles together. So we'll come back and look at that more in detail. So just now I mentioned that if you have a dominant allele, you will see its effects in the organism. And so when we describe those effects of alleles on the organisms, what we're describing is the organism's appearance, its behavior, its interaction with other organisms as an example of behavior, its biochemistry, its physiology, all those kinds of things collectively are called the phenotype. And so the phenotype of this pea plant, the one on the left, the phenotype we would say is it's tall and it's bearing purple flowers, and the one on the right we would say is dwarf or short and it doesn't have any flowers. So we're describing the appearance of the organisms, or the populations of the organisms in some cases. Sometimes we can't see the phenotype overtly, it's not obvious to us, but we would actually need to do some biochemical testing, some physiological testing to understand what the phenotype is at the kind of cellular and tissue level. And so we're going to stick to really simple phenotypes, things like tall and short, red and white, those kinds of things, things which are really, really obvious. Now, the phenotype is affected by two main variables, or two main things. The first thing is obviously the genotype. And so in the case of the pea plants here, the plant on the left is tall because it's got at least, we know it must have at least one big T. So the gene here is the gene which controls plant height. So pea plants are controlled by one gene, or their height is controlled by one gene, and that gene is called T, and it stands for tall. And if you have a big T allele and you are a pea plant, you will be tall. So you can have two big T's, big T, and you'll be tall. Or because big T is dominant to little t, you can have a big T and a little t. We don't know just from looking at this plant. We'd have to do some crosses and some genetics to find out exactly what its genotype was, and this is what Mendel did, or more accurately his gardener did. Now the plant on the right, on the other hand, it must have two little t's. That's the only way we can see the effect of the little t's. If we put a big T in there, so it was big T, little t, the plant would be tall. So if we see a plant that's exhibiting the phenotype associated with the recessive allele, we must know that there are two little t's in the case of the T gene here. So this description of the alleles, big T, or maybe big T, or little t, is called the genotype. Now this is the genotype in regard to one gene. If we were to look at your genotype, and we looked at every single gene you have, and you have 20 to maybe 22,000 genes, we'd have to write out two alleles for every single gene you have. There'd be some exceptions to that, things that exist on the X chromosome, if you're male, but that's what that would look like. So when we talk about genotype in these videos, we're going to be thinking just about one or maybe two genes, so that's going to be no more than two alleles, up to a maximum of 4 alleles in the genotype. So when we get these genotypes, we're often going to present them just as the two alleles, and the uppercase letter will be the dominant allele, and the lowercase letter will be the recessive allele. That's just standard for us to start with. So we have some special names for the different combinations of alleles that we can have. So if the genotype of an organism in respect to the A gene, for example, and these are just examples, there may be an A gene, there may not be an A gene, but you can apply this to all other kinds of genes that you'll come across. If we see big A, we would say that that is homozygous dominant. The dominant coming obviously from the fact that both alleles are the dominant version of the gene, the dominant allele, and homozygous meaning both alleles are the same, homo meaning the same. So homozygous meaning two copies of the same allele in the dominant form. Now if you look at the bottom here, we've got two little a's, so that would be homozygous recessive because both alleles are the same, but they're the recessive allele. And then in the middle there, we've got a dominant allele and a recessive allele. Two alleles again, a dominant and a recessive. And we've got one of each, so that's called heterozygous. Now the mistake we see people making with this verbiage is that they get homozygous dominant, they get homozygous recessive, but then for some reason people like to call heterozygous either heterozygous dominant or heterozygous recessive, and that's wrong. Hetero means two different. And so because it's one of each types of the alleles, we can't call it dominant or recessive, it's heterozygous. Now, whatever the A gene controls, if we've got a big A in the genotype that's heterozygous in the middle there, then we would see the characteristic associated with the dominant allele. But we wouldn't ever say the genotype is heterozygous dominant or heterozygous recessive. We would always say the genotype is heterozygous. Okay, so that kind of wraps up a real introduction to just some nomenclature and some conventions in simple Mendelian genetics. I'll be back and we'll start looking at some more complicated stuff next time.